exploration endeavors. She led the science team for the Veggie hardware validation on space station. I'm sure she's gonna elaborate more on the Veggie. So she led an interdisciplinary group to study the fertilizer and the light impacts on the nutrition and flavor of the veggie grown crops. In addition to the Veggie, she helps with the science needs for other space station hardware and works with the external PIs to get their science to function on station. She also is involved with education and outreach programs related to plants in space. So she has BS in plant science from Cornell University, a PhD in plant biology from Penn State, and a postdoc research from Purdue and Kennedy Space Center. She has worked in the areas of plant space biology and bioregenerative life support. With that, I'm going to pass the time to Gioia.
Thank you, Genwa. I appreciate it. And thank you for inviting me to speak with you all today. I've been really looking forward to it.
Thank you.
So, so hopefully you can all see my slides. And if there are any problems, please interrupt and let me know. And I'll talk with you about our work in space crop production and Veggie. But we'll start with why we're interested in growing plants in space. I love this photograph of astronaut Mike Hopkins. This was the last time he was on space station. He's actually just coming back from his current trip on space station today. And he's showing a Thanksgiving dinner. And so you can see all the food is individually packaged. A lot of it is freeze-dried or thermostabilized. And it's actually a really good diet. There's about 180 different foods and another 20 or so beverages and condiments. But what happens over time is that this food degrades. And so the vitamins in the food degrade and the quality of the food degrades. The organoleptic acceptability will break down. And so it's not a problem on space station. We're sending new food up all the time. But it will be an issue when we, venture farther, when we go for a Mars mission, where the food may have to actually be sent before the astronauts even leave Earth. And so we're looking at crop production now through this sort of narrow lens of being able to supplement this prepackaged diet to provide, vitamins and dietary variety for that mission. And then over time, to grow things. At the same time that you're growing plants for food, there's the potential for psychological benefits of having those plants in that environment where you can't really go outside without a spacesuit. And we're actually collecting data now on potential, behavioral health and psychological benefits of having plants in space. And we've gotten some really great data from the astronauts on that. And then as you all know, plants help to recycle the atmosphere, which is a very important thing when you're in a closed environment of a spacecraft. We have very high carbon dioxide levels in spacecraft. On the space station, it averages around 3,000 parts per million, but that's only with very active CO2 scrubbing. So having plants in an environment where you have, these humans closed inside is going to be really important. And my colleagues have done calculations to show that if you had 20 meters squared of plants, you could actually revitalize the entire atmosphere for one crew member. But at this point, we're working on a much smaller area, but hopefully over time, it will grow into something a bit bigger. And then, water recycling is another area where plants may play an important role in the future. There have been studies that have been done with gray water, with growing plants and collecting the water vapor that comes out of plants through transpiration. And that obviously is a very pure source of water. So plants may have a lot of roles in future space habitations. This is an artist's rendition of what it might someday look like on a lunar base. And this could also be on Mars. You've got your densely packed layers of plants growing inside under electric lights in the same habitat where people are living and working. There are obviously design choices where you could have these in a separate enclosure just for plants, or you could have them in, the primary habitat as is depicted here. Some of the hazards that you would face in this environment can be mitigated by the structure. So you see this very thick wall on this facility that is designed to prevent, the hazards of micrometeorite impacts and to mitigate the hazards of galactic cosmic radiation and solar particle events, which are very real hazards for these environments. Alternatively, you could build the structure underground and just berm regolith over the top of it. And you can see part of this facility is underground. Another hazard of this environment is dust, especially on the moon where there's no atmosphere. Dust is very caustic and abrasive. And if you start to get dust in your equipment, the equipment will start to malfunction pretty quickly. So dust mitigation is a major concern. And that's one of the reasons that we have an airlock, in these facilities, and the ability to keep suits perhaps in the airlock and have suit ports directly into the habitat. So maybe you climb out of your spacesuit and you never bring it inside the habitat. Dust is something that we have a lot of people at NASA working on, dust mitigation, through electrostatic means or other methods. On Mars, the dust is a little less dangerous than on the moon, but it's still pretty dangerous and it can cause a lot of problems. So that's another hazard. A more near-term concept for a plant growth facility might look a little like this, where you have a module that could be launched entirely on a rocket. And it's the same module that I'm sitting inside for my Zoom background. And that module could hook to other modules to form this space habitat. So it wouldn't be, say, a permanently manned base. It may not have quite as much protection for things like solar particle events. And maybe you would have to have a smaller, more well-protected facility for crew to evacuate to in that case. But, this would be a great addition to life in that environment. And, we're already growing plants like this today in some extreme environments on Earth, including the US South Pole Station. So having these modules that could just hook to a facility is something that could definitely be applicable to terrestrial agriculture. So our space crop production vision is to ensure food system security on long durations beyond low Earth orbit. And what I mean by food security is basically, maintaining astronaut health and well-being, and with a diet that's desirable, that gives them all of the nutrients that they need to be able to complete their mission. So for now, we're focusing near term on nutrient supplementation of the prepackaged food. There are specific nutrients that are kind of low in that packaged diet that I mentioned, and there are other nutrients that do degrade over time. Things like vitamin C actually degrades pretty quickly. Vitamin K and thiamine are already kind of low to start with. So those and other elements like potassium are things that we want to focus on enhancing. But then more long-term, we're looking at starting to add caloric replacement to be able to facilitate Earth independence. And so that involves, more of the staple crops. So that gets us to kind of the crop candidates that we're thinking about for this type of a crop production system. And so right now we're kind of on the left-hand side of this chart. We're focusing on salad greens, leafy greens, tomatoes, peppers, radish. These are all things that you can pick and eat fresh without any processing. We do need some way to clean the produce or to ensure food safety, and that's something that is in work, but still needs work. but they can just be added directly to the diet in the form of salads or mixed in with the packaged food. The same with the herbs, and we're starting to do some more work on herbs, looking at, the ability to add these things directly to the diet. The staple crops we break into a couple of categories. So right now we don't have an oven on space station or a microwave. There's really no way to cook anything. There's a small food warmer that gets up to about 180 degrees Fahrenheit, and they do use that sometimes with the produce. And there was an oven that was sent up a year ago to test baking chocolate chip cookies, and they baked one chocolate chip cookie, and it took 2 hours. So you can imagine trying to bake a potato in that. It'd be a good 24 hours for a baked potato, I think. But, it's progress. But because of the lack of convection and some of the other things in the space environment, it's hard to cook things. And so I think we're probably going to be waiting until we have one of these planetary surface bases, like I just showed you, before we have the type of cooking capability. But when we do, adding white potatoes and sweet potatoes would be relatively straightforward. They don't need a lot of additional processing, and they're very, high calorie crops. Things like wheat and soybean, can certainly be grown, and wheat has been grown in space on many occasions, but you need a lot more equipment. You need your threshing equipment, your milling equipment. You need things like, bread machines and pasta makers and tofu presses to go from these raw ingredients to prepared food. And that also requires a huge amount of labor, if you were going to do basic subsistence farming and make all of your food, or more likely automation and robotics, because we're not sending astronauts to Mars to be subsistence farmers. We're sending them to explore Mars. So there's going to have to be a lot of autonomous operations that will be conducted for these types of crops to get them into, ready to eat food. So when we think about plant growth in the environment, I'm preaching to the choir here, but we think about these 5 cardinal factors, temperature, light, moisture, nutrients, and atmosphere, that all have to be within a certain range to grow plants. And in the space environment, these might be a little bit different. So we don't have natural convection unless we have gravity. Without gravity, heat doesn't rise. And so you've probably seen, the candles burning. They show a birthday candle on Earth and a birthday candle in space. And the candle in space is just a small blue dome, because the burning of oxygen is limited by diffusion. There's no up and down, and heat doesn't move and doesn't rise to cause more airflow to make that candle a normal shape like we would see here on Earth. Or the flame of the candle. So we have to have forced convection. We have fans all over space station to distribute the air. We have fans in all of our hardware. Apparently, it's really quite loud up there with all of the acoustic vibrations from the fans, but they're critical for thermal control and atmospheric management. And in partial gravity, we're probably still going to have a degree of forced convection to get kind of normal plant growth. So on the moon, we have 1/6 G. On Mars, we have 3/8 G, about a third of gravity. And so we still don't really understand what we need for those environments. The light may look different, and you all know this, but we might be able to use the light to get specific plant qualities, whether it's yield, nutritional qualities, or even organoleptic or pathogen pest resistance qualities. So those are things that obviously we look to the horticultural community to see where that research is going, and that's driving us to the types of lighting that we're going to need in the future. Fertilizer is something that we have to do more work on. There has been some work on things like composting, leaching, biochar, using fungi to break down those inedible plant wastes and generate, edible processes as well as, fertilizers for the plants. But we have to figure out how to recycle the inedible plant materials and even the human wastes, which tend to be a slightly more disgusting subject for some audiences. But, getting those nutrients back and being able to recycle is going to be critical to off-planet sustainability. And then water is strange in space, and water and air don't mix well. But as we all know, plant roots need water, they need oxygen, and getting that mixture right has been extremely challenging in a microgravity environment. And we really don't understand well how that's going to change in partial gravity on the moon and on Mars. So we kind of group these space crop production challenges, some that are sort of specific to deep space, to the free-floating environment, microgravity, and the strange fluid movement behaviors and the lack of convection. Some that are very specific to a surface environment, such as the dust and partial gravity. And some that are kind of shared between both those environments, how we recycle our water, how we protect from radiation. Pressure is something I didn't really mention, but we're going to be operating a lot of these facilities at reduced atmospheric pressure, and how that impacts plant growth, and how you have to have different partial pressures of critical gases to get good plant growth. But obviously things like transpiration are going to go a lot faster under reduced pressure. And then micrometeorites as a hazard for these different environments. So you have to build these very sturdy, protective structures. Then we have some crop-specific challenges that are the same that are, being faced, for any terrestrial system. How do we increase productivity and stress tolerance? How do we optimize for our environment, or optimize our environment for the crop growth that we want? And how do we manage the scheduling for all of our crops? And then we have some that are really shared, getting very compact plants that'll grow so we can have multiple layers of growth. With this high CO2, food safety is really critical. We don't have great capabilities to test that, but we need to make sure that we're not getting astronauts sick. And, especially in a spacecraft where you have all these human-associated microorganisms just floating around, how do you prevent illness? Getting good nutrient output, especially key nutrients, and just being sustainable when you have all these abiotic stresses, limited vehicle resources, limited crew time or labor, and dealing with wastes. So for now, we're working on the International Space Station, as well as doing a lot of work for this on the ground. The ISS is our orbiting laboratory, and we have how many right now? About 6 people, I think. If the crew one departed, we had 10 or 11, 7 people, I believe we're up to. We've had a lot of astronauts up there recently. The number kind of changes every day, but we've had people up there for more than 20 years, and they're living and they're working. They're doing fundamental biological and physical science research, but they're also testing future exploration hardware that we need, to be able to go longer and farther away. And we can do this in low Earth orbit without, the risk of running out of things, because we can very easily get people home in case of emergency. When we go to the moon, it's not going to be quite as easy. And when we go to Mars, there's not really much of a chance to come back if something bad happens. You're really kind of stuck going out and then coming back. So we need to get things right before we take those next steps. And one of the things that we're testing on the International Space Station is a small plant growth chamber called Veggie. So this is a photo of Veggie when it was first installed on the ISS in 2014. And this is in the Columbus module of the space station, which is the European laboratory. And, the first thing I want you to take away from this photo is what that environment looks like. And just try to imagine if you were living in that environment for 6 months or a year, how important it might be to have something green and growing up there in this very synthetic environment of plastic and metal and cables. It's a very, it's kind of like being on an airplane for 6 months or something. So I think the astronauts seem to really appreciate having some plants there, whether it's for food or for other purposes. And so Veggie is it's a really simple chamber. It uses only about 70 watts of power. It has some, quite nice red, blue, and green LED lights, and it has a fan kind of in the center of the light cap. And those are the only powered components. Everything else in Veggie is manual or passive. So it has these bellows that help to contain some humidity, and it draws air in through the bottom of the bellows from the ISS environment through the plant growth area and then up through that fan, and then it comes out the very top of this light cap. And there are plant pillows, which are essentially grow bags in Veggie, and that's how we contain a solid baked ceramic substrate, controlled release polymer coated fertilizer, and seeds and wicks. And that's how we've been growing plants in Veggie for a while. But Veggie is also a platform, and we can do other types of experiments in Veggie. We've had Petri dish science in Veggie, where we get like about 30 of the 10 centimeter square Petri dishes growing Arabidopsis. We've done magenta chambers. We can do 12 double stacked or even triple stacked magenta chambers in there. And we've done Brachypodium, which is a model monocot. Next month, some of our researchers are going to be growing cotton in some of these tall stacks of magenta boxes. We can do seed germination, and we're looking at other water and nutrient delivery systems that will be tested as well in Veggie. So Veggie is really a platform with some decent lights and a fan. And the bellows are expandable or extendable, which allow you to kind of adjust to get the light level that you want. The lights themselves are also adjustable. And you can keep pace with plant growth by keeping the canopy a set height away from the lights. So it's pretty versatile and very simple. We have another piece of hardware on Space Station you'll see closer to the end of the talk, which is called Advanced Plant Habitat. It's kind of the much more complex big sister of Veggie. So APH, Advanced Plant Habitat, is almost all automated. It has over 180 sensors and control systems and controls almost every aspect of the environment. And it's really more of a plant physiology research chamber, whereas Veggie is a little more like a garden. But it's using the controlled environment of the space station to control temperature, carbon dioxide, and humidity, and remove water from the air. So we've grown a variety of crops in Veggie, mostly leafy green crops. We started with the first test. This was Veg 01A, and here you see astronaut Steve Swanson harvesting the outrageous red romaine lettuce that we grew for this first test. You can see it very clearly, the plant pillows here. They have a quick disconnect that water can be added to, and those little white flaps sticking up in the center are actually wicks that the seeds get planted in. And we had some problems with the first test. Underneath these plant pillows, there's a reservoir that is designed to wick water to the plant pillows. But we found out in the very first test that the wicking rate was insufficient to keep pace with plant growth. And we lost 2 of the plants from this experiment. Steve Swanson intervened and was able to rescue 3 of the plants. We had 1 plant that just failed to germinate at all in a plant pillow due to a hardware malfunction of that. But other than that, the 3 plants that grew, so 3 out of 6 were batting 500, not terrible for baseball. They grew pretty well. We didn't allow the astronauts to eat this first set of lettuce. We needed to do food safety analysis, and so they were returned to Earth frozen. Though they might have had a nibble on a leaf when we weren't watching. This led to the second grow out in Veggie. And so this is sort of a video showing the harvest of the very second test. This is astronaut Kjell Lindgren harvesting. We did a lot better with this experiment. We had 1 plant pillow again with a hardware issue. And so that one didn't germinate, but the other 5 germinated and grew pretty well. The crew were allowed to eat the produce this time. Our food safety analysis came back that it was safe to eat, but we do a prophylactic sanitization where they use a sanitizing wipe, and they place layers of leaves in between layers of wipes in a bag and kind of press for about 30 seconds, and that knocks down the microorganisms on the leaves. So the astronauts got to eat half the produce. We still returned half for food safety and chemistry analysis. We also are doing microbial community analysis with this, but you can see here that they were pretty excited to eat it. Everybody toasts their lettuce at home, right? So they seem to like it. That's Kimia Yue, Kjell Lindgren, and Scott Kelly. And Scott and Kjell actually made some speeches. This was kind of a big deal for us. And so Scott talked about the logistical complexity of having people live and work in space for long periods, and the supply chain that's required, and how we have to be more self-sustainable with our food supply when we go to Mars. And Kjell, who's a physician, talked about the benefits of eating the fresh food, the contribution of the plants to the ecosystem of the space station, and the psychological benefit of seeing green growing things in that environment. So it was really great to hear them, talk about all the things that we'd been thinking about for a while. After that, they wanted to grow some more plants. And so we had a set of plant pillows up there with zinnias. And we'd sent the zinnias because we weren't entirely sure what order the plants would be grown, and we wanted them to have a non-edible crop to grow while we were getting the food safety data. But we also wanted to test flowering and see how well flowering worked in veggie for, obvious reasons for future fruit production. But we had some issues with the zinnias, as you can see here. And the issues actually turned out to be a ventilation problem that was unrecognized. When power is lost to the rack, which is the hardware that powers veggie and supports it, and it was turned off for an unrelated reason. But when it's turned back on, the veggie unit appears to come back on, the lights come back on, all the indicators come back on, the indicator for the fan comes back on. But what we found out later is that the fans don't actually come back on. So it seems to be a little bit of a software glitch. They're easily resettable if you know that the problem exists. But at the time, we didn't know that the problem exists. And so the fans were off in veggie for several weeks during this experiment. And we started to see some very strange symptoms, as you see here. We saw the epinasty of the leaves, some beautiful guttation droplets along the edges of the leaves. They were still watering the plants. And, the water was just building up because it wasn't going anywhere because it was limited by diffusion. We saw really strange, stressed out plant growth. You'll see in the right-hand side of this. In the back, you can't really see too well, but the plants tended to be very stunted. And in the front, they were sort of just trying to grow quickly to outgrow all of this waterlogging, but the growth itself looks really abnormal. And then right around 3 days before Christmas in 2015, we get a call at 4 a.m. because the space station is on Greenwich Mean Time that something funny is growing on the plants. And so you see here this sort of gray fuzzy leaf. There's some more gray fuzziness in other spots. There were these water-soaked spots on the plants, and a fungus, which ended up being Fusarium oxysporum, basically opportunistically, just colonized these plants and took over. And Scott Kelly went in, we talked him through, we developed procedures for cutting off damaged tissue, cleaning things up, resetting the fans to high. They had been going to set the fans to high earlier when they noticed all the water, but they had an emergency spacewalk, so it postpones, plant science. So they reset the fans, he got things cleaned up, and he tried really hard to rescue the plants, but we actually still had several of these plants die. So only 2 actually ended up surviving this experience. So it was an extremely stressful time for our team. I don't think Scott was too happy with us. But then, he got the plants to start flowering and everything changed. It went from being this really stressful, awful experience to being, something really beautiful and wonderful. And so you see here in the left-hand photo, that's Scott Kelly, who was our 1-year astronaut, and Misha Koryenko, who was the 1-year cosmonaut. And they're celebrating 300 days in space, which was a really big deal. They took photos at 100 days, 200 days, and 300 days in space. And you see Scott holding the flower. He took it to the cupola, which is the window of the space station, to get some natural light. And they had flowers on the dining room table one night. And these are called profusion zinnia. So we had two that survived of two different colors. It was a mixed variety. And they actually produced a profusion of flowering. They just continued to bloom and bloom and bloom. We ended up with about 28 flowers on these two plants at 90 days, which was when Scott harvested. He asked if he could harvest on Valentine's Day instead of the day after, which was a Monday. So it was his day off. And we said, of course. And then he asked if he could make a bouquet. And we said, sure. And so you'll see that bouquet at the end. I forgot to include it here. But that was really pretty awesome to have, the first ever off-planet flower arrangement. And so it ended up being kind of an exciting and happy experiment after all the earlier stress. So after that, we've had additional crews growing in Veggie, and we've added additional crops and additional harvest techniques. So here Shane Kimbrough is showing off the first time we tested cut and come again harvesting, repetitively harvesting from the same lettuce plants. We got a lot more produce from that really productive lettuce variety. We added a second veggie unit, and here Scott Tingle's showing off some mixed cropping. We've got red lettuce, Waldman's green lettuce, and Mizuna mustard in there, though he left kind of a few dead leaves on the Mizuna, but he seemed to really enjoy growing the plants. And we've sent some new crops. So we sent red Russian kale, dragoon lettuce, wasabi mustard, a nice spicy mustard green, extra dwarf pak choy, a little baby bok choy, and amara mustard. That one's also called Ethiopian kale, or we call it steak plant because it's got a very meaty flavor. We've grown these in different combinations with different tests, and actually just a week and a half ago, the amara mustard and the extra dwarf pak choy were just harvested on Space Station. So these were grown for 64 days with multiple harvests, and the astronauts made some really interesting foods out of them. And two of these were crops selected with an education outreach program that we're involved with called Growing Beyond Earth, where we have middle school students and high school students in classrooms around the country. We have around 250 schools right now. That have analog growth chambers that are similar to Veggie in their classrooms, and they're collecting crop selection data and providing those data to NASA. So if you have a crop variety that does well in 50 different classrooms where you've got different, humidity and temperature, air conditioning or no air conditioning, high altitude, low altitude, and, we have some students who forget to water and others who water too much. If a crop variety does well in all of those different environments, it might be a good candidate for spaceflight. And so we then select those crops, do higher fidelity testing in our controlled environment chambers at Kennedy Space Center to see how they grow under ISS conditions. And if they do well, then they go to space. So it's been very exciting for us and the students involved. We've heard from the astronauts how important aroma is and how much they like smelling the plants. When Mike Hopkins was harvesting recently, he said it smells like the produce aisle of the grocery store, which apparently is much better than the space station normally smells. So I think that is another aspect that connection to Earth becomes really important. And we've seen them rate the veggie plant growth very highly in terms of sensory stimulation for, vision, taste, and odor, aroma. We've done Veggie-4. So this was the experiment where we looked at the different red to blue to green light ratios on Mizuna mustard crop growth. And we also assessed nutrient composition. We did taste tests with the astronauts on orbit. So we're looking at organoleptic appeal, and we have a big focus on microbial food safety. And this is when we started the assessments of crew behavioral health. So we're still working on processing all these data, but this has been a very successful set of experiments. We had Veggie-4A that was just a 30-day grow out, and then Veggie-4B that was a 60-day grow out with multiple harvests. And we got, a lot of great feedback of the astronauts adding the fresh produce to their diet and trying all different things. They get all the different foods out and then they try different things. We got a lot of crew engagement in this because we really wanted to make sure that the astronauts who were, rating how how the plants impacted their well-being got to actually participate in growth. So we had some pretty good photos of happy astronauts, and that's really why we're doing this, is to enhance that. Recently, we've also grown radishes on space station. This was an experiment by Professor Carl Hassenstein at University of Louisiana Lafayette. This is in the plant habitat. So you can see it looks a little bit different than Veggie. It has a somewhat larger growth chamber. Veggie is 0.13 meters squared. The APH growth area is 0.2 meters squared. This is Mike Hopkins again. He's got a huge green thumb. He really has enjoyed growing the plants, and so he got up there and started with the radishes. Sorry. This is the system that has about 180 different sensors and control systems. So you can see some of those here. I'm not going to talk too much about it today, but if you're interested, we can talk about it in the questions, because these would be systems that you guys could all propose to do research in if you're interested through NASA research announcements. We've had another couple of interesting experiments this year. We had some very old plant pillows. They were up on space station for more than 2.5 years before the astronauts had time to grow them. We normally plant 3 seeds in each pillow. And we had this one, it was a mixed test with red Russian kale, extra dwarf bok choy, wasabi mustard, dragoon lettuce, and outrageous lettuce. And none of the lettuces germinated very well. So Mike Hopkins decided to try to transplant some of the seedlings from the brassicas that had germinated well into other pillows. And you look at the seedling here on the left, and he got almost no roots, when he was pulling. It was already 10 days old. The pillow is sort of got a gasket, so it was very hard to remove, and the roots broke off. And we thought, that's never going to survive. But this is one case when microgravity and the weird water behavior actually worked in our favor. And so you see here the original on the left and the transplant on the right, and it survived, and it did well. So it was a little bit behind the original, but we were very surprised that that plant was able to survive with such small amounts of roots. So that was a really interesting lesson learned from this recent test. We've also tested seed handling in microgravity. We've developed a water-soluble, food-safe seed film that uses a polymer the seeds are embedded in. And we had astronaut Hopkins plant these plant pillows. So the plant pillows were sent up there empty. We didn't have seeds glued in like we do with the others. And instead, he planted the seeds on orbit to test this technology, which was very successful. You see the lettuce here on the left-hand side. We got extremely good growth, actually faster lettuce growth than we'd seen in earlier tests from the seed film. So that was something else that we just tested in February of this year, January and February. And then most recently, as I mentioned, Amara mustard on the left and extra dwarf pak choi on the right were grown for 64 days. They had 4 harvests, and you see that the extra dwarf pak choi even started to flower, and Mike Hopkins got in there with a paintbrush and was pollinating the flowers. So we had some really good experiments. We're getting some plant samples back on the next rocket that comes back, and we look forward to analyzing and getting those results out. So I'm just going to end with a few slides on things that we are thinking of that plant breeders and genetic engineering might be useful for space plants. And a lot of these, I think, would apply for other types of controlled environment plants. We have a lot of different gaps and needs that we need to figure out before we're ready for Mars. But coming up with ideal crops is definitely one of them. And so these targeted areas fall into a number of different categories, and I'll just go through them really quickly. We think there's a lot of potential for plant growth and development, coming up with a very compact plant like this pepper over here, which is only, a little bit taller than the 4-inch pot that it's in, that has high yielding. So obviously we can select some of these from available crops, but these aren't things that are being developed for field agriculture. but they are going to be suitable for controlled environment agriculture and certainly for space. So we want a high edible to inedible biomass ratio. We want reliable germination, rapid growth to first yield, uniform growth between individuals, propagules with long stability, long shelf life, whether they're seeds or cuttings, and ways to keep those viable, especially under conditions that might not be optimal, like radiation and different temperatures. We want sustained production capability over a long duration. So, repetitive harvesting, fruiting for a long period. Low debris formation is going to be really important in microgravity and also in indoor ag, especially vertical farms. You don't want a source where disease could get in. But you saw with the zinnias, one of the reasons they were selected is they didn't shed a lot of pollen. They didn't shed petals. We tested all of these things before we selected those. And having that stuff remain attached even when the plant, senesces those tissues is going to be very important. We're also interested in a custom microbiome, maybe of plant protective and plant growth promoting microorganisms, so that we can keep the plants healthy and keep the crew healthy. We have aspects of plant physiology that we're interested in having targeted, things like stress tolerance, especially water stress. We go between kind of a flood situation and a drought situation really often in space. Ability to grow well under all of the unique conditions that I talked with you about earlier, high CO2, we have low humidity, we've got a really pretty uniform temperature because it's all like human associated environmental conditions. If we can have plants that grow well under short photo periods and save lighting energy, that could be really good. And plants that are just tolerant for things like broad environmental range, reduced pressure, no dormancy, low aroma, low release of volatiles. So all of the things that we just don't want building up in that environment. And if we can get plants that have a high sodium tolerance, that's going to be really important in the future when we start to recycle things like urine. Urine has a lot of good minerals for plant growth, but it has sodium, and getting that sodium out can be very tricky, though we obviously, are working on it and thinking about that. But a preference for ammoniacal nitrogen is also going to be something that could be beneficial for those types of plants. In terms of nutrition, we don't want to change things too much, but we would love to enhance some of the things that we need. The antioxidants, beneficial phytonutrients, lutein and zeaxanthin are really important because eye health is often compromised in spaceflight. And then some of the minerals that are either low or break down in the diet. And we don't want a lot of anti-nutrients in our produce. And then, organoleptic acceptability, having strong flavors and good textures, it's really important. It can be the healthiest thing in the world, but if it doesn't taste good, they're not going to eat it. And we want to make sure that they can taste things. And a lot of times there's a bit of a fluid shift in microgravity. So astronauts say it's a little like having a head cold. So they like spicy things. You see, big bottles of sriracha all over their table. So strong flavors could be really good. And then post-harvest, there's some really interesting things to think about there. Shelf life is obviously important, reduced processing, but easy digestibility of the inedible plant material. So, having the ability to maybe have less lignin and less structure in the plants so that it breaks down more quickly, because we don't need to stand up against gravity, right? So we don't need all that structure. Maybe we can even have more food and less structure. We also are thinking about how we could use the inedible plant waste as resources for other things. So I mentioned edible fungus earlier, could do fish, but then there's even cellular agriculture, using it as feedstocks or scaffolding for things like synthetic meat or dairy or eggs. And then there may be other useful materials that we can produce from the inedible plant material, such as shielding or fuels. So I'll just stop there with the picture of Scott Kelly's bouquet, and just want to thank you on behalf of our teams at Kennedy Space Center. We've got a lot of people involved in this. Scientists, plant scientists, molecular biologists, microbiologists, chemists, and then,
engineers
of many different varieties, as well as all the people that just get things done. And then, of course, the astronauts and our funding sources. So I'm going to stop sharing and take any questions.
Super cool talk. I'll ask questions.
Keep sanitized before being brought up into space, because I know some plant pathogens can be seaborne. So
yeah, we do sanitize the seeds. So we surface sterilize them. We try to buy seeds that are clean to start with, but we surface sterilize them. And that's something that we're actively working on. Mostly we've done, dry methods, because the seeds do get stored for a very long duration dry. And so we use like a chlorine gas fuming. But we also test, to see the immediate germination, the stored germination, and then what grows out of those seeds in terms of microbial contaminants. One thing that we haven't done yet, but we're interested in, is working with some more of the seed companies to try to, really ensure clean seeds. We've mostly been purchasing our seeds. We're doing this, pretty small scale. And so we'll just go to Johnny's or, some seed catalog to get the seeds. But I think in the future, it's going to be a lot more, working with growers who can really ensure seed cleanliness, because the endophytes, the ones inside, I think, are what we're more concerned about. Great question.
I have a question. You mentioned at the beginning about this subsistence farming versus the exploration activities. And that's one observation that I also thought about when we had a NASA project with Dr. Fred Davis one time. And so the question was, how is that you're going to get this balance? Because obviously when people, all the things that you showed us gives us the impression that people are spending a lot of time doing this.
Yeah.
So what is the target time that you believe would be a balanced time for exploration to Mars, right? What is the target?
So our target is 0 man hours. We want to have everything automated with the option that the crew can then participate in things that they want. They can either turn off the robots or whatever for things that they want to do. I think there are always going to be activities that they want to do. Like they may always want to pick the ripe strawberry or the ripe tomatoes, right? Because that's fun. That's what we want to do. But they're probably not going to want to manually water the plants or do the inspection. I think we're going to probably have remote support. We'll be getting photos back to Earth. We might be able to advise, but we're looking a lot at plant health monitoring using things like hyperspectral or multispectral imaging, volatile organic, chemical sensing, and other approaches to really be able to detect the stress, and then using machine learning or AI to mitigate that stress once it's detected before it starts to really impact plant growth. So ultimately, we want a system that could be entirely, automated, and the astronauts can then do things when they want to do it, but not because they have to. Mm-hmm. Yeah. Well, that facility unfortunately was shut down in Houston, short-sighted budget cutting. And so, we've done a lot of work. We have the calculations. So we know that, if you're growing 20 square meters of pretty much any plant, you're going to be able to generate enough oxygen for one crew member and completely recycle the atmosphere for one crew member. If you grow 40 to 50 square meters, you can do at least calories for one person. It's going to take a while before we get to that point. You know, as you can see, we're growing 0.13 meters squared or 0.2. So we're much smaller scale at this point. We're really focusing on supplementing the packaged diet with the idea that when we get to a location like the moon or Mars and people are staying longer, then we'll start to kind of grow the system. And we've had some, we have some folks working on things like autonomous greenhouses or deployable greenhouses that could be, on the surface of Mars or the moon that could just start to supplement that. But I think the engineers are all, it's so interesting because they're very squirrely about using biology for life support, even though when the biology fails, it's usually due to an engineering failure, not a biological failure. So, we've worked very hard to try to educate them, like if you keep your engineering good, the plants are going to grow fine and they're going to do their job. But there's still work going on in other countries as well. So Europe has a program called the Melissa Project, which is Microecological Life Support System Alternative. They're doing a lot of work with algae as well as higher plants and cyanobacteria and other microorganisms. So different groups are doing different things. But right now, our main focus is, because we can't provide the diet for a Mars mission, our main focus is supplementing that diet. We know we can do the air and the water and other things through physical chemical means, uses a lot of power, requires a lot of, consumables, but it can be done. But we can't make food through any other way at this point. So that's where we're focusing. Good question, though.
Well, there's a question in the chat box, and maybe you just answered it, about is NASA in particular interested in research on food that's derived from microorganisms and their byproducts?
Yeah, that's a great question too. There's a group out at Ames Research Center, which is in California, and they are the synthetic biology leads for the agency. And so they've looked at different microorganism approaches for food and for other things, making fuel, making, feedstocks for different purposes. So I think that synthetic biology is certainly an increasing field. We've had some meetings with the cellular agriculture folks who are doing like synthetic meat or dairy. So there's definitely interest. We're personally not doing a lot of work on that at Kennedy Space Center. Our focus is really on the plants, but we're keeping an eye out and looking at how we can interact with those groups.
Okay. And now there's
another question, if it's okay.
Go ahead, Sarah.
Yeah, I just wanted to ask about what you use to sanitize the lettuce leaves before consumption. Maybe that was like kind of a makeshift solution, but I was wondering if you anticipated that.
Yeah, well, we sort of hedged our bets. We had sent, so there'd been work through a NASA small business grant for a company that developed this produce sanitizing solution called Prosan, which is a citric acid-based solution. And so usually it's used like you take the powder, you mix it in water, and they use it on like naval submarines. You go into a strange port, you can dump your vegetables in this, and it knocks down a lot of the microorganisms of concern. So we worked with them and we've developed a wipe that has that solution in the wipe. And we sent it because it's also used for food processed surfaces and hardware, because we didn't want any non-food safe materials coming into contact with the chamber. So we had the wipes available for cleaning the hardware. And then we had started testing them when we thought, okay, we're going to be able to, have them eat the lettuce. We tested them with this sort of, press that we call it the Prosan press, where we have leaves in between layers of wipe. It's very labor intensive. For lettuce, it's not too bad, but for like Mizuna with all those little leaves, it takes the astronauts a long time to do this, even if they're only pressing them, in the thing for 30 seconds. So we're definitely interested in alternatives from a produce sanitizing approach. We've had a company that's worked on some of this. And so, I think it has to be something that doesn't damage the quality of the produce, because then they're not going to do it. It has to be something that's going to be low labor and low consumables. So I know some companies have looked at UV. UV is hard with plants because there's a lot of corners that microbes can hide in that are blocked by the light. Ozone might be an option,
I
peroxide.
recently learned about bacteriophages as
Cool.
in that application, and that could perhaps like save the good bacteria for the gut health of the astronauts.
That would be very interesting. Yeah, I think the microbiome is something that we're really just getting into, and I think that there's a lot of potential. There's a fungus that one of my interns got really excited about called Muscador albus, and that one produces like this combination of 16 volatiles that then sanitize and kill stuff. And so, I think bacteriophages could certainly be a really interesting approach if you can make sure you get all the ones that have, E. coli, salmonella, staphylococcus, all those bacteria of concern and kill them. That'd be great. Cool.
There's another question in the chat box. How do you regulate your atmosphere, particularly the partial pressure of oxygen, ethylene, and CO2 and water in the plant chambers?
Yeah, that's a great question, Hannah. In Veggie, we don't do any regulation. So Veggie is just taking the atmosphere of the space station. The space station atmosphere itself is a controlled environment. And they have, a number of volatile scrubbing and different ways to scrub CO2, generate oxygen. But for Veggie, we're just seeing, can we grow in that atmosphere? For advanced plant habitat, we have a carbon dioxide injection system. We have, as I said, it's much more complicated. We have these porous ceramic elements that regulate humidity. So they, depending on the temperature, you can get water to condense on them and remove humidity, or to add moisture to the air. You can change fan speed and control temperature through different ways of heating and cooling. And then we have potassium permanganate to scrub out ethylene for that chamber. So that one's sort of the all bells and whistles. Veggie's the no bells and whistles. But actually, we seem to be able to grow plants in both. There used to be problems several years ago in the space shuttle and in other space facilities that had very high levels of ethylene and some other volatile organic contaminants, which caused plant growth problems. They have since worked pretty hard to clean up the air in these environments. And so far, knock on wood, we haven't had too many weird issues with the air quality on space station. So we're not sure in the future if we're going to be in a more closed system or a more open system. But as we reduce the pressure of spacecraft, we're doing 0.8 atmospheres or 0.75 atmospheres for future exploration spacecraft. At a certain level, and I can't remember the exact level, but my colleagues know this, you actually start to have to increase the partial pressures of CO2 and O2 to compensate to get normal plant growth. But I think that's when you're taking things down really pretty low pressure. I don't think at 0.8 atmosphere, you're going to have too much of a problem. But we probably won't be pressurizing any plant growth facilities because that would be way hard. Yeah.
And we have another question about how active is the research on growing plants on Mars or on the moon as compared to the research that you're currently doing in a microgravity environment on the International Space Station?
Yeah, that's an excellent question. It's one of those, it's a little farther down the road, so we have less attention on it. So we're thinking about these things, but we're not actively developing hardware. Once you get a little bit of gravity, some of the challenges go away, we think. We work pretty closely with the fluid physicists, on the water delivery challenge. And we're trying to develop kind of an omni-gravity water nutrient delivery system that will solve some of the problems that we've had with water, because water's just it's been really challenging. And so I think if we can get something that will work in all different gravities with water, that'll be great. I'm not going to hold my breath on that, but we're hopeful. The other alternative that people have talked often about is using regolith, which is the dirt, on Mars or on the moon, as a way to grow plants. There are some minerals in these substrates, at least the Mars rovers are sending back things like they found nitrates on Mars. So there are some good potential things there, but then there are also some, decremental things like there's planetary wide perchlorate on Mars. So that material would have to be remediated through a process that would take out the perchlorates. And you can do that with microorganisms and through other methods before you would grow the plants. And we have some partners at different universities who've been doing testing with simulated Martian regolith. You might amend that with something like a compost, perhaps. The moon is another interesting challenge. I'm actually part of a team that's looking at maybe trying to do a small plant growth experiment on the moon. The lunar regolith, the lunar material, it's just such a fine particle. It basically turns into concrete when you add water. So figuring out how to get that, so that roots can actually penetrate, it's an interesting challenge. I personally think that hydroponics is probably going to be a lot easier, even with the nutrient management issues, trying to take it, the inedible plant material and reclaim those nutrients. I think hydroponics is a little easier to control, but I'm certainly not ruling out the potential for using regolith. And so we have sort of small research going on in those areas, but most of the things that we're trying to build, we're hoping will work in various different environments. So I think that's kind of an answer to your question.
Okay. And I'm going to read one more question from the chat, and then we'll let you go today, because I think it's about that time. But the question is, are there any experiments in the pipeline to begin studying ideal microbiomes for growing plants in microgravity? And where can we learn about university labs you collaborate with?
Yeah, those are really interesting questions. The microbiome is so fascinating. And, I think we're sort of doing kind of a multi-pronged approach to that. We are keeping an eye on the companies that are doing like biostimulants. There's a number of companies out there. It's really challenging to work with NASA if you're a company, because it doesn't go through the same grant pipeline. So we sort of are keeping an eye on what's going on in the industry for biostimulants. But at the same time, we're analyzing the microbiomes that are coming back from our plants that have been grown in space. We identify if any of the microbes that are there are, have plant growth promotion potential. And so we're working with, trying to kind of get a little bit deeper on that. We actually recently hired a woman who has a great background in microbiome research. And so she's trying to take that a little bit further. And yeah, Emmerich is also working on that. So definitely contact him. There are a number of people in universities that, are working in this area. And so I think we're not all in yet, but we're really actively watching what's going on in that area, because I think developing kind of a custom microbiome for the space plant is a really great target, something that will help protect the plants, protect the crew, promote their growth. I think that's all going to be really interesting. And probably Emmerich knows a lot more about this and could get you a lot more information. But in terms of other labs doing NASA research, if you just Google like different NASA research announcements in space biology, you can see who's gotten the grants and is working in this area. And you can find out more about it. You can also get in touch with me and I can put you in touch with people. So
wonderful. Well, I want to thank you again for an excellent presentation. It was a great way to end our semester seminar series. I know we've had some other speakers on, and so I want to thank everybody who's participated. I think that I hope that the department and all of our visitors have enjoyed this controlled environment agriculture spring seminar series. And this is the last one. We'll see everybody again in the fall, hopefully, and we'll certainly stay in contact, Dr. Massa.
Great. Thank you. Thank you all for coming and all the great questions. Have a wonderful evening.
Thank you. Bye now.